Hello, my name is Carol Leibel, and today we want to examine the structure of cells, part 2. When we left the last PowerPoint, we were examining the structure of the mitochondrion. Now we want to examine the structure of plastids. Plastids are found in plant cells and some smaller protist cell. There are two basic types of plastids. You have your chromoplast, and chromo means color. So these plastids contain some sort of pigment. This chromoplast right here contains anthocyanin, which causes the plant tissue appear to be red. The plastid you're probably most familiar with are chloroplasts. Chloroplasts contain chlorophyll, which causes the plant tissue to appear to be green. Now, leucoplast or amyloplast are plastids that are responsible for storing starch. So if you were to examine some white potato tissue underneath the microscope, you would notice that it has lots of leucoplast. So let's examine the structure of the chloroplast. Chloroplasts have two membranes, outer membranes that are nice and smooth. The third membrane is involved in forming structures called thylakoids. A stack of thylakoids is called a granum, or plural is grana. So what you're looking at right here and also right here is a granum. It looks like a stack of dimes. Each one of those dimes then is a thylakoid. Inside the thylakoid is where you're going to find chlorophyll and other pigments involved in the process of photosynthesis, where light energy is converted into chemical energy. So now we're going to examine the structure of a thylakoid. A thylakoid has an area where we'll find pigments involved in photosynthesis, and you'll also find other electron carriers forming the electron transport chain. In the lumen of the thylakoid is where water is split that is needed for the process of photosynthesis. So the light reaction is taking place in the thylakoid. Now, outside the thylakoid is a substance called the stroma. Inside the stroma, you're going to find enzymes involved in the light-independent reaction that has the ability to make that carbohydrate, or you probably know it as glucose. Now, you might be asking, where in the world or how did these organelles evolve? One of the advantages of being a eukaryotic cell is that these membrane-bound organelles can specialize and do certain functions. So it allows you to divide the work throughout the cell. How did they ever evolve from a prokaryotic cell? Well, I want you to remember about the endomembrane system. So here it is real fast. Remember, the endomembrane system includes the nuclear membrane, the endoplasmic reticulum, the Golgi apparatus, the plasma membrane, and lysosomes, okay? And so what they think happened is over time is that there was a folding in of the plasma membrane. And as it folded in, it became the nuclear envelope. And that nuclear envelope then gave rise to the Golgi apparatus and the endomembrane system. So that's how some organelles evolved. Now, other organelles evolved through a process called endosymbiosis. And this would include the mitochondrion and the chloroplast. What they believed happened is that you have this prokaryote, and through the process of phagocytosis, it engulfed an aerobic heterotrophic prokaryotic cell. Now, for whatever reason, it did not digest or break down that little prokaryote, and that little prokaryote took up residence inside the larger cell. Now, remember, it could do cellular respiration and produce ATP for the cell. Over time, some of the genes from this little prokaryote was transferred to the nucleus. And so it no longer was an independent cell and became an organelle we know as the mitochondrion. Now, they also believe this process evolved in the formation of a chloroplast. The same sort of thing happened. Phagocytosis took place. It took up residence inside that cell. The photosynthetic prokaryote could do photosynthesis and make carbohydrates. Some of the genes from that prokaryotic cell eventually were transferred over to the nucleus, and so it no longer was an independent prokaryotic cell, and it became the chloroplast. Now, the evidence that we have that endosymbiosis took place to form the mitochondrion and the chloroplast is that inside both of those organelles, we have circular DNA like a prokaryotic cell. Inside both of those cells, we have ribosomes, and those ribosomes are smaller like prokaryotic ribosomes. They do photosynthesis like a prokaryotic cell, and they divide like prokaryotic cells. So all the evidence points to the fact that these organelles, once upon a time, were independent prokaryotic cells. There's some evidence now that the flagella and cilia also evolved through this process of endosymbiosis. So now let's examine another organelle, and that includes the ribosomes. Now, ribosomes are workbenches for protein synthesis. Here is an electron micrograph, and we can see that ribosomes can be free-floating, just found floating in the cytosol, or they can be attached to the endoplasmic reticulum. Ribosomes are made from ribosomal RNA and proteins to form two subunits, a large subunit and a small subunit. Those two subunits are only together if the process of protein synthesis is occurring. Cells can have thousands of ribosomes. The next little organelle that we would like to examine, or it's not an organelle, is the cytoskeleton. Now, the cytoskeleton is found underneath the plasma membrane and throughout the cytoplasm. It gives shape to the cell. It is also involved in the movement of the organelles and cytoplasmic streaming found in plant cells, and provides support and structure for the cell in general. There are three types of structures found in the cytoskeleton. You have your large microtubules, and then you have your smaller microfilaments. And in between the two, you also will find a class of intermediate filaments. So this is a nice little video that shows the cytoskeleton and things being moved on the cytoskeleton. Kind of cool. So what about microtubules? What are they made out of? Well, microtubules are made out of little protein spheres called tubulin, and two of them come together to form something called a tubulin dimer. And if you think about a string of pearls around a hollow tube, that's a microtubule. Microtubules are involved in the formation of cilia and flagella, so movement. Other proteins can attach to them, motor proteins, and they also give a tremendous amount of support for the cell. So we're going to see this little video that shows you how microtubules are assembled and disassembled in the cell. And I got to say, the speaker doesn't work very well on this. The little movie comes a little bit further down the line. I am so sorry, so please forgive me. I just had a moment. So let's talk about the structure of cilia and flagella. One of the things that you'll notice is that you will find here 9 doublets of microtubules. So that's 9. And then you find 2 singletons in the middle. So this has a 9 plus 2 structure, if you will. Both cilia and flagella are made the same way. And here, this represents the plasma membrane. So it's actually coated, if you will, in plasma membrane. There are these little protein arms called dynein proteins, and they are responsible for actually kind of moving up and down the microtubules, causing them to bend in one direction or another direction. Now, these flagella and cilia are always attached to a structure called a basal body. Down below, this is a structure of a basal body, and it's made out of 9 triplet microtubules. And there are no microtubules in the center, but that is what the cilia and flagella are anchored. If you were also to look at the structure of a centriole, the structure of a centriole is identical to that of a basal body. So now here comes the movie. And as I said, let's just watch it, and I'll go ahead and I'll talk you through it. So basically, what that little movie was saying is that microtubules are assembled and disassembled within the cytosol of the cell, and that they're made, as we mentioned, from these little tubulin dimers, and they are only assembled and disassembled at either one end or the other, not at both ends. So here is a short little video showing you some flagellated cells and how fast the flagella can beat. And over here is a much longer flagellated cell. And here we have some paramecium that is ciliated, and chlamydomonas that is also ciliated. And you can see how that the difference between cilia and flagella happens to be the number and the length of the structure. As we mentioned before, these structures are both covered with the plasma membrane. And here are those little dynein arms. And they kind of have this ability to walk up and down those microtubules. And as they start to walk up and down the microtubules, they cause them to slide past one another, causing them to bend. The difference between the flagella and cilia is the length and the number. Flagellated cells will have 1 to 8 flagella. Ciliated cells can have thousands of these structures. Also, flagella are longer than cilia, and so as they're moving, the flagella kind of moves in a wave-like structure, whereas the cilia kind of moves like the oars in a rowboat. The centrioles are microtubule organizing centers in animal cells, and they're found in an area called the centrosomes. Plant cells have centrosomes, but they do not have centrioles. And we've already mentioned the structure of the centrioles having 9 triplet microtubules, just like the basal bodies. All right, so now let's talk about microfilaments. Let's go back. Microfilaments are much thinner than microtubules, and they are solid. They're not hollow, and they are actually made out of 2 intertwining strands of actin subunits. Microtubules are responsible for this process called cytoplasmic streaming that you can see in this little video, causing the organelles to move around within a plant cell. So they're also involved in muscle contraction. So here is a unit involved in the contraction of a muscle, and up here These are the actin filaments, and these are the myosin arms. And as these little heads flip, it causes this sarcomere, if you will, or this unit to contract and to move in, causing the muscle to contract. This is a nice little diagram showing you this process of cytoplasmic streaming and also how a cell can move with a pseudopod. So again, microfilaments are a part of the cytoskeleton, and more specifically, we found that actin filaments are important in muscle cells in the contraction of the sarcomere or the contraction unit of a muscle cell. They're also important in a pseudopod changing shape and moving like an amoeba. And they are also important in cytoplasmic streaming that's found in certain plant cells. As we have mentioned, something that's in size found between the size of a microtubule and a microfilament are the intermediate filaments. They are a collection of different kinds of protein structures. Their main function is to help maintain the shape of the cell, the anchorage of the nucleus and certain other organelles, and the formation of the nuclear laminella. So let's look at this nice little video about
the cytoskeleton is a network of protein fibers that determines the shape of cells without cell walls, provides for the movement of cells, provides for the structural support and movement of organelles within the cell, and provides the framework for moving and separating chromosomes during cell division. The three types of protein fibers that make up the cytoskeleton are thick microtubules, thin microfilaments, and medium-sized intermediate filaments. Microtubules are hollow tubes assembled from subunits made out of the protein tubulin. Subunits not forming microtubules are dissolved and scattered throughout the cytoplasm. As subunits are readily available, microtubules can be assembled and disassembled as required by the cell. Subunits are assembled into microtubules by microtubule organizing centers found at various locations within the cell. All eukaryotic cells have a microtubule organizing center near the nucleus, called the centrosome. During cell division, the centrosome produces a football-shaped array of microtubules called the spindle apparatus. It separates chromosomes and divides them between the two new daughter cells. In animal cells, a pair of organelles called centrioles are located by or in the centrosome. Centrioles are short, barrel-shaped rings with 9 microtubular triplets forming their outer edge. As a cell prepares to divide, the centrioles are duplicated. One pair moves to each daughter cell. In cells that have structures called cilia and flagella, the centrioles multiply, move to the surface of the cell, and they become anchored just beneath the plasma membrane, forming basal bodies. 10 pairs of microtubules grow out of each basal body. One pair grows from the center of the basal body, while the other 9 pairs form the outer infrastructure of the cilia or flagella. A gap, the width of the microtubule, exists between each of the outer microtubule pairs. Numerous motor proteins firmly attached to one pair of microtubules have legs, cross the gap, and can walk on the adjacent microtubule pair. The force applied by the motor proteins causes the microtubules to bend. The synchronized movement of large columns of motor proteins attached to microtubule pairs trying to walk up or down adjacent pairs creates the rowing motion of cilia, wave-like motion of flagella. Cilia, Latin for eyelash, are short and numerous structures that extend from the cell membrane. Cilia move single-celled organisms such as Paramecium through their environment with a rowing motion. Flagella, meaning whip in Latin, are usually considerably longer in cilia and fewer in number. Flagella move in a wave or whip-like motion. The sperm of most animals and algae rely on flagella for movement, as do a number of single-celled organisms such as eukaryotes. Microtubules are also believed to be involved with the movement of organelles, such as vesicles, within the cytoplasm. One hypothesis holds that vesicles and other organelles have receptor proteins to which motor proteins attach and then walk an organelle to its destination via the network of microtubules that extends throughout the cytoplasm.
So most cells have materials external to the plasma membrane, except for plant cells, meaning they don't have a cell wall. Cell walls are found in prokaryotes, plant cells, fungi, and some protists. The cell walls in plants are made out of cellulose, but in fungi they're made out of a modified carbohydrate called chitin. In prokaryotes, they are made out of peptidoglycan or muramic acid, and the cell walls found in protists can vary depending upon the species. Now, a plasmodesmata is the channel found between plant cells, and it allows for direct flow from cytoplasm to cytoplasm in adjacent cells. So what you're looking at right here, here's the cell wall, okay? And then right here is the plasmodesmata, and it allows this direct connection between adjacent cells. Remember, cell walls are not alive. Cell walls are dead, and basically they give structure and they prevent the cell from bursting when they are put into a hypotonic solution. The middle lamella, by the way, is the material, kind of like glue, holding these cells in place. Now, in animal cells, you do not have a cell wall. But what they do have is they have an extracellular matrix, or it's called a glycocalyx. The extracellular matrix is made out of glycoproteins like collagen, proteoglycan, and fibronectins. These glycoproteins are connected to a receptor protein in the cell membrane called integrins. They're basically the extracellular matrix is used for support, adhesion, movement, and identity. So if you're looking at this, here is the inside of the cell membrane, all right? And then this is an integrin, and it's anchoring here the glycocalyx. And this may be indeed a protein glycan, all right? And again, these things may also be used for adhesion or identity, identifying different kinds of cells. Animal cells, like plant cells, have ways to communicate to one another through different kinds of junctions. So plant cells actually have three different types of junctions. One is called a tight junction. And a tight junction are neighboring cells that are basically pressed together. Think of it kind of like a rivet, if you will. Now, desmosomes are anchoring junctions, and they fasten cells together into strong sheets. And then you have your gap junctions, and gap junctions are communicating junctions. These are the junctions that are really like the plasmodesmata, and they're channels between adjacent cells, and they allow for direct contact. So in looking at this nice little illustration, here are the gap junctions, and you can see there are channels that will allow the cytoplasms to be in direct contact. These are the desmosomes, and I kind of like to think about desmosomes kind of like shoelaces in a shoe, in which they're kind of holding the two sides of the shoe together. All right, and then here are those tight junctions that are kind of more like rivets. So we have a couple videos that give you a nice review about.
Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
All right, so the next slide is going to show you a nice little review of a plant cell.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. All plant cells have mitochondria, just like animal cells do. Sugars produced by photosynthesis are broken down and converted to ATP in mitochondria. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
So I hope you now have a fundamental understanding of prokaryotic cells and eukaryotic cells and all of their little organelles. It's really quite amazing. So have a great day and we'll talk to you later. Bye.